Hi, I'm Vladimir and I'm at the Czech National Supercomputing Center IT for Innovations. Coming up, I'll introduce the Salomon and Awesome supercomputers and briefly explain how to assemble a genome. So, this impressive machinery here is the Salomon supercomputer that I've been running my computing jobs on during the summer. It has 1,000 computing nodes, each powerful on its own, but to efficiently utilize a supercomputer, you need to be able to distribute the problem to as many nodes as you can. Now, the bioinformatics software that I was using showed this very well, because we're assembling a genome of a whole species, and it requires massive amounts of processing power and memory, but it can be neatly divided to multiple nodes. So assembling DNA has only become practical recently thanks to next-generation sequencing technologies. Basically allows us to obtain large amounts of DNA fragments and kind of try to stitch them together to form one single DNA sequence. And we're dealing with large amounts of data here, and we're using a special file system for that called the Lustre file system. And it's convenient because it can be distributed and read in parallel. And the uses for assembling a genome can be, for example, personalized medicine, where you give treatment based on a person's DNA constitution. Software I've been using, Abyss, is just one of the assemblers out there. But reading papers on it was fairly fascinating, so it's my project of choice. It assembles DNA sequence from a lot of fragments, as mentioned before, and it does this by representing them with a de Bruijn graph. In the Bruijn graph, every node is one fragment, and nodes differ by one DNA letter only. That means they highly overlap, and that they likely belong on the same place on the final sequence. You can then start merging the nodes together one by one until you get one node in the end, and this node represents the final sequence. This is the flowchart of the whole Abyss pipeline. It's fairly complicated, so we're only going to focus on the part at the end, which is this. As a clarification, the rectangles with double borders represent stages or processes in this flowchart, and arrows just show the data flow between them. And we're only interested at this output here, the scaffolds. Scaffolds are not continuous DNA sequences. They contain gaps in them, and we know the length of these gaps, but we don't know the DNA information they should contain. And moreover, there's more scaffolds than just one at the end. Part of Abyss, one of its stages, is called Sealer. It's the one I've been working on. It processes these scaffolds and tries to fill in the missing data. Now the problem with Sealer is that it takes a very long time. This graph shows the node usage of a node that runs the Sealer job. After the initial part, where it builds a required data structure, which is a multi-thread part, you start sealing gaps with only one thread, which is why the node is heavily underused. Ideally, you would want most of this white space to be filled with this blue kind of color, which would indicate the threads are running, which in this case they're not. So I multi-thread this with OpenMP, and this is what we get. Way more used node. This is the initial part with building the data structure, and then all the threads start sealing the gaps. It kind of starts declining here, which is because some threads finish their work before the others. I took this a step further and optimized it with MPI. In MPI, every node gets a different color here, so after they build a required data structure, each of the nodes starts sealing gaps. So as you can see, most of them, actually all of them, are being almost 100% used. Ultimately, we're interested in execution time comparison. So this green column is the original sealer, and it takes 561 minutes. Now compare that to OpenMP version, which lasts only 55 minutes. And there's the OpenMP plus MPI version, which lasts 29 minutes. Now, MPI is not that much faster in this case because there's a bottleneck here. Some gaps simply take much longer to close than the others, and they determine the minimum amount of time for the job. The MPI program was run with 8 nodes, each with 24 cores. So the results of the summer project are the following. 2 pull requests were actually accepted into the original project, one of which is the OpenMP parallelization of Sealer, and the other one is a utility that writes sealed gaps to a file, which helps you debug Sealer while it's running. There were also 2 failed attempts at improving performance, one of which is a file reader in Abyss, and the other one is improving performance of building the required data structure that I mentioned before, both of which only improved performance by several %, which was not sufficient. I would like to thank Martin Mokrich for introducing me to bioinformatics and guiding this project, Ben van der Valk and Sean Jackman from Canada's Michael Smith Institute for reviewing my changes and giving me tips, as they're the original creators of Abyss, Martina Kovarova and Barbora Polakova for helping me record this, and of course, Summer of HPC, Technical University of Ostrava, and IT for Innovation Supercomputing Center for having me here and providing the required infrastructure. Thank you for watching.